Have you ever wondered how biologists are able to capture stunning images of cells, tissues, and organs at a microscopic level? The answer lies in the powerful tools of image acquisition, which allow researchers to visualize and study the intricate structures of living organisms in the incredible detail. In this video, we'll take a closer look at the fascinating world of image acquisition for biology and explore the key concepts and techniques that make it all possible with a real-world example. What is image acquisition? Image acquisition is the process of capturing digital images of biological specimens using various imaging devices such as microscopes, cameras, and scanners, aiding biologists and other life scientists to study the intricate details of cells and tissues to understand their functions. There are a few basic concepts to understand when it comes to image acquisition. First is resolution, which refers to the number of pixels in an image. The higher the resolution, the more detailed the image will be. Second is magnification, which is the ability to make an object appear larger than its actual size, which is important especially when studying tiny cells and structures. Third is contrast, which is the difference in brightness between different parts of an image. This is important for highlighting specific features in the specimen. Finally, there is color, which can provide additional information about the specimen, such as the presence of certain pigments. There are several types of imaging techniques used in biology. One of the most common is bright field microscopy, which uses visible light to produce an image of the sample. Another technique is fluorescent microscopy, which uses fluorescent dyes to label specific structures within the sample. When the sample is illuminated with a specific wavelength of light, the label structure emits a different wavelength of light that can be further captured by the camera. Confocal microscopy is a technique that uses a laser to illuminate the sample at a specific depth. The microscope then captures the emitted light only from the specific plane, allowing for a 3D reconstruction of the sample. Advanced imaging techniques have emerged in recent times, such as super-resolution microscopy and high-throughput microscopy. Super-resolution microscopy surpasses the diffraction limit of conventional optical microscopy, enabling researchers to visualize structures with much higher resolution than previously possible. On the other hand, high-throughput microscopy allows rapid acquisition of large amount of image data, aiding in visualization and quantification of biological features on a larger sample size. Before imaging can take place, the sample must be prepared. That involves treating samples prior to imaging in order to enhance contrast, remove unwanted debris, or modify the sample in a way that makes it more amenable to imaging. This often involves staining or labeling the sample to highlight specific structure. The sample is then loaded to the microscope for imaging. Now that we have covered the basic concepts of image acquisition for biology, let's take a closer look at a real-world example. In this experiment, me and my colleague are investigating the dynamics of neutrophils following injury in a 3-day-old zebrafish embryos using a high-throughput imaging setup. By capturing images of the neutrophils over time, we aim to gain insights into the underlying biological processes at play. We begin by screening the embryos for a positive GFP signal and select those that exhibit the desired activity for further imaging. After screening, the selected embryos need to be prepared for live imaging by choosing an appropriate mounting medium that will provide the necessary support and stability to the sample while minimizing any potential stress or damage. For this experiment, we loaded the embryos onto a 96-well plate with agarose grooves to maintain their position over time. And once they are loaded, the embryos need to be oriented correctly according to the experimental requirements, which in this case involve lateral orientation. Post-mounting the embryos, the 96-well plate was sealed to prevent any potential evaporation of the media during the course of imaging. Once the plate was sealed, it was loaded onto the microscope for imaging with the incubator to ensure that the physiological conditions of the sample remain constant throughout the experiment. Finally, after loading the plate into the microscope, various imaging parameters such as the laser intensity, exposure time, camera settings were optimized to achieve the best possible imaging conditions for the sample being imaged. Post-optimization, we initiated the image experiment and began acquiring images of the live embryos. Finally, we successfully acquired high-quality images of live zebrafish embryos post-injury using a high-throughput imaging setup. I hope that this session has provided you with a general overview of the concepts and components of image acquisition and their practical application in the field of biology. In our next session, we will be discussing image processing tools which play a crucial role in analyzing and interpreting the large volumes of image data generated in modern biological research. Thank you for your attention. I look forward to see you in our next session.